Would you believe me if I told you that 1 in every 16 vertebrates was a catfish? And you may be wondering, well, what's so great about catfish? This order of ray finned fish may not cross your mind very often, but their wide diversification, especially in tropical freshwaters, have marveled evolutionary biologists in recent years. This makes these organisms ideal for studying rates of diversification as well as cryptic diversity, which occurs when species that are evolutionarily distinct appear very similar in appearance. This is important for understanding species' evolutionary background so that effective conservation methods can be implemented. I am currently standing in the Natural History Museum, which holds over 8,000 species of catfish from all over the world. In a study conducted by Day et al., the diversification and biogeography of catfish in the Congo Basin was analysed using nuclear DNA sequence phylogenetics. Phylogenetics refers to the practice of determining the evolutionary relatedness of individuals and placing them in trees of life, such as how family trees are constructed. The study tested the rates of diversification as well as both the origin of groups and the species richness within a particular genus, Chiloglanus. So imagine you're a researcher heading off to the Congo Basin in an attempt to survey these crazy catfish in the hot summer sun. While taking samples of either fin clips or muscle tissue, you may come across species with long whiskers, known as barbels, used to sense their surroundings, or perhaps brightly coloured catfish with elegant stripes or spots. Once you've gained your samples, you then need to analyse them in the lab and create phylogenetic time trees, which show how organisms originated through time. As you can see, there is an amazing amount of visible diversity within this group. However, what was uncovered by the researchers was the sheer amount of cryptic diversity within the Chiloglanus catfish. They found an incredible 80% increase in the species richness, which could be one of the greatest discoveries of unknown diversity in vertebrates in recent times. The researchers hypothesized that the fast-flowing freshwater environment may constrain the traits of these catfish, leading to convergent evolution and thus diversity that is hidden beneath their similar outwards appearance. To understand convergent evolution, imagine the invention of a hat to protect yourself from the harmful sun rays. Perhaps in the Sahara Desert, someone has come up with the same solution to protect themselves from the sun. Neither of you have passed this innovation to each other in any way, but instead you've come up with the same solution to a similar environmental pressure. Such is the same with catfish. Different species have adapted similar traits due to similar environmental constraints rather than relatedness. This study found that the family of catfishes Makrokiddae have a constant rate of diversification, compared to the usual pattern of a fast increase and then slowing down. This general model seen elsewhere is indicative of adaptive radiation, when a large amount of free niches are taken and eventually filled up. However, in the tropics, a constant rate has been seen for several animal species, likely due to greater niche space and opportunity. The species may also have not yet reached their carrying capacity, which is the maximum amount of individuals of a species that an environment can support. The Congo Basin has proven to be a major area for constructing diversity due to its stressful environments. This is exceptionally highlighted in the Chiloglanus catfish, in which their stressful, fast-flowing environments have created less competition, hence allowing them to exploit this niche to a high degree. Who knew catfish had so much mystery to them? However, with every study, there are always limitations. Cryptic diversity by nature can be very difficult to detect, especially when using molecular phylogenetic trees, which could incorrectly show gene flow as cryptic variation, as seen in previous studies with frogs. Gene flow is the transfer of genetic material from one population to another, such as through migration, and thus this error could lead to larger estimates of cryptic diversity than are actually present. From our whistle-stop tour around the Congo Basin, we can see that catfish are a crazily diverse group of organisms. This wide diversification is due to the stressful environments of the catfish, as well as rich niche space that allows for a constant diversification rate. So maybe the next time you visit your local zoology museum, take a look at the catfish section, and you never know what new diversity you might uncover.